The Constitution of India, Preamble. We, the people of India, having solemnly resolved to constitute India into a sovereign, socialist, secular, democratic republic and to secure to all its citizens justice, social, economic and political, liberty of thought, expression, belief, faith and worship, equality of status and of opportunity, and to promote among them all fraternity, assuring the dignity of the individual and the unity and integrity of the nation. In our Constituent Assembly, this 26th day of November 1949, do hereby adopt, enact and give to ourselves this Constitution.
Hello children, how do you do? Hope you are healthy and happy. Dear children, today onwards you are coming to the school to collect your answer paper, and with that you will be showing your notes also. Dear children, don't worry when you are showing your notes, because if you have not completed your notes or if you are not up to date in your notes, you should not take any tension. Till where you have completed, you have to show, and the remaining portion you can do slowly and complete it. Okay, be always happy. Dear children, today we are going to learn and understand the new chapter, chapter 9, Photosynthesis. Already we know what is the meaning of photosynthesis. In this chapter we are going to study about photosynthesis in deep. So we can move to the chapter. It is the physico-chemical process by which green plants use light energy to synthesize organic compounds. What is photosynthesis? It is a physico-chemical process where the green plants, they are using light energy for synthesizing the organic compound. What do we know about photosynthesis? We know that chlorophyll, light and carbon dioxide, these are the main factors or components required for photosynthesis, which we have already understood and studied in our previous class. The experiments to prove these are as follows, that is chlorophyll, light and carbon dioxide are the important components of photosynthesis. Related to them, experiments are there to prove. You can see which are those experiments. Number 1, variegated leaf experiment, and number 2, half leaf experiment. In the first case, that is variegated leaf experiment, you can see the figure where you can see this is the variegated leaf before it is undergoing starch test. And in this leaf, we can see two areas. One area where you can see green color, the other part is with yellow color. The green color is having chlorophyll in it, and yellow color, chlorophyll is there, still we can say it is colorless. That is, chlorophyll means the content of chlorophyll is very minute in those areas. So we can say it is nil in those areas which is yellow in color. Now when this leaf is taken for starch test, when the leaf is being done for starch test, we can see after the test what happened to the leaf. You can see the yellow color, it is as such, it is not affected by the starch test, it remains colorless. But the area where we have seen green color, it is being affected by starch test, and the area becomes blue-black in color. So from this we can prove that chlorophyll is an important factor needed for photosynthesis. I think you have understood this experiment. Now experiment number 2, half leaf experiment. In this, a part of leaf is enclosed in a test tube containing potassium hydroxide soft coated. That is, in the test tube, the half part of the leaf is being inserted and this leaf will be having the cotton which is having KOH in it. That is potassium hydroxide, and we know that potassium hydroxide absorbs carbon dioxide. Now, the other half of the leaf is exposed to air. The other part of the leaf is exposed to air, that is, it is out of the test tube. Only the half part is being inserted into the test tube. Now, the setup is placed in the light for some time, and that apparatus which is used there is being placed there in the light for some time. Now, that leaf which is exposed to the light is tested for starch. Now, that leaf is taken for starch test. The exposed part of the leaf tested positive for starch. Now, the leaf which was exposed there, that is, which is not dipped into the test tube or which is not been soaked by potassium hydroxide, that part of leaf has given a positive test when a starch test is conducted there with that leaf. And what happened to the leaf which has been dipped there, you can see. The portion that was in the test tube tested negative for starch. This proved that carbon dioxide is required for photosynthesis. Why the portion was tested, that is why the portion that was in the test tube tested negative for the starch? Because already we said that potassium hydroxide is present there in the test tube, and the part of leaf which is inserted in the test tube that contain carbon dioxide, surely what will happen? Photosynthesis has not taken place because potassium hydroxide absorbed or it used to absorb carbon dioxide. That's why it tested negative. I think you would have understood this is the apparatus used for half leaf experiment. Here you can see a bottle or a, we can say, a bottle is being taken there in which half portion of the leaf is dipped there, and in the bottle KOH is present there, and again the half part is being out of the bottle and is exposed to the sunlight. Later on, this leaf is being taken for starch test, where we can see that this portion tested negative for starch test and this portion tested positive for starch test. So from this we can say that carbon dioxide is an essential factor for photosynthesis. Okay, my dear children, that is the second experiment. So both the experiments are very, very important. Now next one is early experiments. In the early experiments, again many scientists have discovered many things related to photosynthesis. We can see number one, Joseph Priestley. He took candle with bell jar and mouse for the experiment and concluded that air is necessary for the growth of a plant. He discovered the fact that plants restore oxygen in the air. Joseph Priestley, he conducted experiment with the candle which is being placed in the bell jar and a mouse is also there. And first experiment he conducted with the bell jar in which candle and mouse was there, and later on he found that mouse was dead because the candle which was lighted there was releasing carbon dioxide and there is no fresh air which is able to clear the air, polluted air in the bell jar. Now later on he took mint plant, he placed mint plant in the bell jar with a lighted candle and he placed mouse in that bell jar and he found that mouse was alive, the candle was also lightning there, burning there, and the plant is also fresh there. It shows that oxygen is being evolved from the plant and carbon dioxide which is being coming out from the respiration of mouse as well as from the burning of candle is being absorbed by the plant which is present there in the bell jar. That's why it has been said that Plant restores oxygen in the air and air is necessary for the growth of a plant. Understood? And the next scientist was Jan Ingenhousz. He conducted the experiment with aquatic plants in light and dark, and he concluded that sunlight is essential for plant processes that purifies the air. So he conducted the experiment with aquatic plants, and he said that sunlight is an essential factor for photosynthesis. Next, Julius von Sachs. He said that green parts of plant make glucose and store as starch. He done the experiment and he concluded that in the plant there is a green part which is helping to prepare glucose and it is being stored in the form of starch. Then T.W. Engelmann, he splitted the light using prism into 7 colors, that is Vitriol. And he took green algae which is called Cladophora and he placed in a suspension of the aerobic bacteria, and the bacteria were used to detect the cycle of oxygen evolution. So again here it is being again concluded that oxygen is an important factor for photosynthesis. Engelmann, what he has done in the experiment, he took the light using prism and this light was splitted into 7 colors, and he also took green algae Cladophora, and this green algae was placed in the suspension of aerobic bacteria, and later on he found that bacteria were used to detect the signs of O2 evolution. Isn't it? Next one is related to the other scientist, that is Cornelius van Ney. He did experiment with purple and green bacteria and demonstrated photosynthesis is a light dependent process with hydrogen from water reduces carbon dioxide to carbohydrates. He concluded that oxygen comes from water and not from carbon dioxide. Finally, the correct equation for photosynthesis was discovered, and it was set as when 6CO2 combines with 12H2O, glucose is formed there with the formation of water as well as oxygen. So this was the ultimate conclusion given by the scientist. So Cornelius van Ney, he said that photosynthesis is a light dependent process where hydrogen, that is he said that process with hydrogen from water reduces carbon dioxide. Isn't it? Water reduces carbon dioxide to carbohydrates, and later on he said that oxygen is coming from water and not from carbon dioxide. And last, it was concluded with the equation of photosynthesis that when carbon dioxide and water are combined together, glucose is formed there with the formation of water and oxygen. So this is the early experiments conducted by certain scientists on photosynthesis. All these are important, my dear children. You have to go through it. I told you in the beginning, Joseph Priestley, he performed the classic bell jar experiment, and he observed that when lit candles are placed in a closed container, they soon go out. But if a living plant was put in the container, the candle would stay lit longer. So he was the first person to show that plants produce oxygen gas. So Joseph Priestley was the first person who said that plants are producing oxygen gas. Isn't it? So, my dear children, you can see here this is the bell jar where in the first experiment we used lit candle as well as a mouse. But later on what happened? The candle was off as well as the mouse was dead. But in the second experiment, when we used the green plant with a lit candle and mouse, living mouse, what happened? 3 of them are living there. So from this, we concluded that air is important factor for the process of photosynthesis. This is again important. Now next we can see where does photosynthesis takes place. It has been said that photosynthesis is an important process taking place in plant. Where this photosynthesis is taking place, we can see photosynthesis takes place in green leaves. So we know that in plants, stem, root, leaves are the main part, flowers are the main part, but among these, the green leaves are doing photosynthesis. Now cells of green leaf contain chloroplast. The green leaf which is present there in the plant, it contains chloroplast. You can see the structure of chloroplast. Here you can see these are laminae and they act like the skeleton of the chloroplast, maintaining the distance between thylakoid. Isn't it? The thylakoid is present this one, and each thylakoid they are having in between the laminae. The laminae they are acting like a skeleton and they are maintaining the distance between the thylakoid. Next one, you can see here the stroma region. It is the space outside the thylakoid membrane and is the location of Kelvin cycle in which organic compounds are formed such as glucose. So in stroma, dark reaction is taking place. In the stroma, already in 9th standard we have studied, there are 2 reactions taking place during photosynthesis. One is light reaction, the other one is dark reaction. The dark reaction is taking place in stroma and there is a space which is seen outside the thylakoid membrane. Now granum, you can see this is granum and it is a stalk of thylakoid and the plural of granum is grana. And the grana we know that light reaction is taking place. The stroma is to dark reaction, grana is to light reaction. Now this you can see here again photosystem. Clusters of pigments and proteins that absorb light energy are found in the sac-like photosynthetic membrane called thylakoid. So this is thylakoid where the clusters of pigments are present there and these are absorbing light energy for doing the light reaction. So this is thylakoid, this is lamina, this is stroma and each stack is grana. Okay. So I think you have understood the structure of chloroplast. Now next one related to photosynthetic pigments. Pigments are the substances that have the ability to absorb light at specific wavelengths. So what are pigments? They are substances which are having the ability to absorb light at certain wavelengths. And the pigments in plants are divided into 2 groups. Number 1 chlorophyll, number 2 carotenoids. The first one is chlorophyll. They are green pigment and chlorophyll A is bright or blue-green in color. Chlorophyll is green color pigment and chlorophyll A. Among chlorophyll there are different types of chlorophylls we can see in the plants. They are chlorophyll A, chlorophyll B like this. Different chlorophylls we can see. And among those chlorophyll A is bright or blue-green in color. Now chlorophyll B, they are yellow-green in color. Now carotenoids, they are yellow-orange in color. Carotenoids, they are yellow-orange in color. So these are the different types of pigments we can see in the process of photosynthesis. They are helping to do photosynthesis. Now next one, the role of various pigments in photosynthesis. We have to see what are the roles played by these various pigments. Chlorophyll A, it is the major pigment involved in photosynthesis. It absorbs blue and red regions of the visible spectrum. In the visible spectrum, we know that the geode there, it is absorbing blue and red region. Isn't it? That light it is absorbing and it acts as reaction center of light harvesting complex. In the photosynthetic process, there is a light harvesting complex, and in that complex chlorophyll A is acting as a reaction center. Understood, my dear children? Now all other pigments except chlorophyll A are known as accessory pigments. We understood and we know that is chlorophyll A is a main pigment which is doing photosynthesis. Other than chlorophyll A, the other pigments which are present there is helping chlorophyll for doing photosynthesis, and those pigments are called accessory pigments or antenna pigments. They are known as accessory pigments or antenna pigments. Now we can see the function of accessory pigment. Accessory pigments absorb light energy and transmit it to chlorophyll A. So what is the function of accessory pigment? They are absorbing light energy and they are transferring this light energy to chlorophyll A, as chlorophyll A is acting as a reaction center in the light harvesting complex during photosynthesis. They protect the chlorophyll A from photooxidation. That is, in the presence of light, oxidation process is taking place, and this is being blocked by the accessory pigments present there. So, in that manner, they are protecting chlorophyll A from photooxidation. Now, the next part is related to the next topic, that is absorption spectrum and action spectrum, which is connected to photosynthesis. First one is absorption spectrum. A curve obtained by plotting the amount of absorption of different wavelength of light by a particular pigment is called absorption spectrum. So what is absorption spectrum? It is a graphical representation where the amount of absorption of different wavelength of light is being placed and it is being absorbed by a particular pigment. So the maximum absorption of light, or we can say the maximum absorption of different wavelength of light by a particular pigment which is being plotted in the form of a graph, that is called absorption spectrum. This is the graph related to absorption spectrum. Here you can see graph plotting absorption of light by photosynthetic pigment is on y-axis and it is against the wavelength of light on x-axis. So this is x-axis and y-axis. So the wavelength of light is on y-axis and the amount of light absorbed is on x-axis. And you can see here chlorophyll A is there, chlorophyll B is there, and carotenoid is there. And the maximum light is being absorbed by chlorophyll A in the region of 400 nanometer as well as in the region of 700 between the 600 to 700 nanometers, isn't it? So from this we can say that the maximum absorption is being taking place by a particular pigment such as chlorophyll in the region of red region, or we can say in the blue region of the visible spectrum. This is important. Now next one is action spectrum. It is a graphical representation showing the rate of photosynthesis at different wavelengths of light. So what is action spectrum? The action done during the process of photosynthesis. Here it is said that the different wavelength, isn't it, of light is there, and in the different wavelength of light, how much photosynthesis is being done, that is being plotted there in the form of a graph, and that is called action spectrum, isn't it? So here the rate of photosynthesis is on y-axis and the wavelength of light is on x-axis, isn't it? Now it has been said the rate of photosynthesis is on y-axis and the wavelength of light is on x-axis. So this is x-axis and that is y-axis, where we can see the graphical representation of the absorption of maximum light by the pigment and the photosynthesis is taking place there. Here again you can see in the region of 400 nanometer as well as in the region of 700 nanometer the maximum photosynthesis has taken place. Okay, my dear children, I think you would have understood about absorption spectrum. In the case of here absorption, amount of light absorbed, that is, this is the y-axis and here the wavelength of the light, this is the x-axis, where we are able to understand that the specific pigment is able to absorb the maximum light energy, that is absorption spectrum. And in the case of action spectrum, again you can see the rate of photosynthesis where it is maximum is the action spectrum. So that both the graph and both the term action spectrum and absorption spectrum is very important. You have to study it. Now the graph showing action spectrum of photosynthesis superimposed on absorption spectrum of chlorophyll A. So here we can see a graph where we can see the absorption spectrum as well as the rate of photosynthesis, that is action spectrum, is shown here, and from this we can understand what in the region of 400 nanometer as well as in the region of 700 nanometer the maximum absorption is being taken by the pigment and the maximum rate of photosynthesis is being done there in the same region, that is 400 nanometer as well as in the 700 nanometer. Understood, my dear children? So that is how the graph is showing the action spectrum as well as the absorption spectrum of chlorophyll A. It is again important. Now we can see what is the difference between absorption spectrum and action spectrum. In the case of absorption spectrum, it shows the amount of light absorbed at different wavelengths. And action spectrum, it shows the use of light energy of different wavelengths in photosynthesis. So here the amount of light absorbed is absorption spectrum. Here the amount of light is used in different wavelengths during photosynthesis, that is action spectrum. In the case of absorption spectrum, it is the graphic representation of wavelength absorbed by a pigment. In the case of action spectrum, it is a graphic representation of the rate of photosynthesis at different wavelengths of light. Now here, in the case of absorption spectrum, it requires a single exposure to light having intensity required for photosynthesis. In the case of action spectrum, it requires study of light utilization at various wavelengths. So that is the difference between absorption spectrum and action spectrum. Now the next topic is light harvesting complex. The minimum number of pigment molecules participating in a light reaction is called light harvesting complex. It is also called LHC or photosynthetic unit or quantazones. What is light harvesting complex? It is the minimum number of pigment molecule when participating in light reaction. The light reaction, minimum number of molecules are participating, and that is called light harvesting complex. And it is also known as photosynthetic unit or quantazones. The photosynthetic unit consists of a reaction center and a light harvesting pigment molecules. In the case of photosynthetic unit, we can say it is having two areas. One is reaction center, the other one is the light harvesting pigment molecules are present there. Now the first one is reaction center, which is also known as photocenter. It consists of special chlorophyll A molecule which traps light energy. So reaction center, which is also known as photocenter, is having a special chlorophyll A molecule which is trapping the light energy. Now reaction centers are of three types. Number one, P700, and that is seen in PS1, that is photosystem 1. So P700 is photosystem 700 and it is seen in photosystem 1 reaction center, where chlorophyll A molecule with an absorption peak around 700 nanometer is taking place there. That is, in PS1, chlorophyll A molecule will be absorbing the light around 700 nanometer of wavelength. That's why this center is called PS1, that is photosystem 1. Now number 2 is P680, which is seen in photosystem 2, and here the chlorophyll molecule will absorb the light energy around 680 nanometer. As 680 in the 680 nanometer region, the chlorophyll molecule is absorbing the maximum light. That's why that system is called photosystem 2. Now the third one is P870, and is seen in bacteria, and bacterial chlorophyll molecule with an absorption peak around 870 nanometer. So the chlorophyll present in the bacteria, they are able to absorb the light energy in the wavelength of 870 nanometer. That's why this reaction center is known as P870, or we can say it is seen in the bacteria, where the center is known as P680. Sorry, P870. So there are 3 types of reaction center we can see. Number 1, PS1, which is having P700, the absorption peak, and number 2, PS2, that is photosystem 2, where the absorption peak is P680, and the third one is P870, where the absorption peak is 870. Now the second one, so that is the first one related to the photosynthetic unit, and number 2 is light harvesting system, which is also known as antennae. These are pigment molecules which absorb light energy of different wavelengths. So in the case of antennae, the antennae, it consists of the pigment molecules which are able to absorb light energy at different wavelength, and it transfers the energy to the neighboring pigment molecule. So from the molecule which are present there, they will be absorbing the light energy, and from that molecule, the light energy is transferred to the nearest molecule. And next one, finally, all the pigment molecules transfer their energy to the reaction center, and in that manner, from one molecule to another molecule in the antennae, it is being transporting the light energy from one molecule to other molecule, and like that, this light energy is being transported to the reaction center, to the chlorophyll A, where chlorophyll A will be trapping it, and it will be using this light energy for doing photosynthesis. Now, the photochemical reactions take place in the reaction center. Photochemical reaction. So, when the energy, or we can say when the light energy from the nearest molecule is being trapped, and when it is being transferred to the reaction center where chlorophyll A is present, there what is happening there? Photochemical reaction is taking place, isn't it? In that reaction center, photochemical reaction is taking place. Now we are moving to the next topic, that is mechanism of photosynthesis. So till now we understood that there are certain area present in the green leaves which are helping for doing the reactions such as light reaction and dark reaction. We are just now understanding about light reaction, where we understood grana is doing light reaction, and in the light reaction, light harvesting complex are present there, which are also known as photosynthetic units or quantazones, and these photosynthetic units, they are having two areas. One is reaction center and the other one is antennae, or we can say it as light harvesting molecules. And now we are going to understand about mechanism of photosynthesis. Photosynthesis involves two phases. One is photochemical phase, which is known as light reaction, Other one is biosynthetic phase, that is called dark reaction. Understood? So the mechanism of photosynthesis, it involves two reactions, dark reaction and light reaction. The light reaction is called photochemical phase and the dark reaction is called biosynthetic phase. The first one is photochemical reaction. This is light dependent phase in which ATP and NADPH are formed in the presence of light. My dear children, this area is very important. Photochemical reaction is the first phase in photosynthesis and it is light dependent phase as the name photo means light. So this phase is light dependent and during this process ATP and NADPH are formed there, which are very important factors for the process called dark reaction. Understood? Now it takes place in the grana of chloroplast. That is, photochemical reaction is taking place in the grana of the chloroplast. Light reaction consists of the following steps. Number 1, light absorption. Number 2, splitting of water. Number 3, oxygen release. And number 4, photophosphorylation. These are the steps related to light reaction. And the first one is light absorption. Number 2, splitting of water. Number 3, oxygen release. And number 4, photophosphorylation. Now, next one, the two pigment systems which are involved in light reaction are photosystem 1 and photosystem 2. Already we understood about this photosystem 1 and photosystem 2, isn't it? Now, photolysis of water, that is, it is the light including splitting of water, that is photooxidation during photosynthesis. This is again important. So in the light reaction, photolysis of water is taking place where in the presence of light, the splitting of water is taking place during photosynthesis, that is called photolysis of water, and this results in the release of H+, that is proton, and electron and oxygen. When photolysis of water is taking place, that time a proton, electron and oxygen is evolved or released. The splitting of water is associated with PS2, that is photosystem 2. The splitting of water is associated with photosystem 2. I think you have understood about it. Now next one is phosphorylation. The process by which ATP is synthesized by the cells, isn't it? In the case of mitochondria as well as in the case of chloroplast, in these organelles ATP is synthesized and the process through which ATP is synthesized is called phosphorylation. What is photophosphorylation? It is a synthesis of ATP from ADP and inorganic phosphate in the presence of light. Photophosphorylation means in the presence of light, the synthesis of ATP is taking place from ADP and inorganic phosphate. What is ADP? It is adenosine diphosphate. So, adenosine diphosphate and inorganic phosphate, when they will combine together in the presence of light, ATP is synthesized and ATP means adenosine triphosphate is formed there. That is called photophosphorylation. Now, we have to understand the two types of photophosphorylation is taking place in the plant during photosynthesis, that is during light reaction, and that is non-cyclic photophosphorylation and cyclic photophosphorylation. So, photophosphorylation is of two types, non-cyclic as well as cyclic. The first one is non-cyclic photophosphorylation. It occurs in the lamellae of grana. In the grana, lamella is present there, and there the non-cyclic photophosphorylation is taking place, and during this process, the loss of electron from photosystem 1, that is the reaction center of P700, is compensated by the electron from PS2, that is the reaction center P680. The PS2 gets electron from water. So here what is happening? There is a loss of electron taking place in PS1, and that loss of electron is compensated from PS2, and how it is being compensated from the water, that is water splitting, or we can say photolysis of water is taking place in PS2, that is photosystem 2, and from there the electron is released, that is being given to PS1, that is photosystem 1. It involves the following steps, that is non-cyclic photophosphorylation involves the following steps. Number 1, reaction center chlorophyll A of PS2 absorbs light of 680 nanometer. So what is the first step? That is the reaction center chlorophyll A molecule is present there, and it is absorbing the light energy of wavelength 680 nanometer. And when it is absorbing that light, that time chlorophyll A molecules will be getting excited, and because of that the electrons present in them will be excited. Okay. Now these electrons are picked up by electron acceptors. So when the electrons will be excited, surely it will be jumping from its original state to the higher level. So those electrons, they will start to jump from its original state to the higher level, and when it is jumping, that time it is being picked up by the electron acceptor. So here this is non-cyclic photophosphorylation. It is, as the name indicated, non-cyclic, that is, it is not in the form of a cycle. That's why it is known as non-cyclic photophosphorylation. So we can see here what is happening, isn't it? In photosystem 2, that is P680 nanometer area, this is the reaction center, and what happened here, the chlorophyll A molecules, they are absorbing the light energy, and because of that they are being excited, and the electron jumped from that center, and this was being accepted by the electron acceptor, and the electron from that area is moving to the cytochrome complex, and this complex is having the electron carrier which are able to carry electron from one part to another part during that process, and from there the electron is reaching to the photosystem 1, that is having P700 nanometer wavelength of light, and there also simultaneously chlorophyll A molecule is present, which will be absorbing the light energy, and from there the electron as usual will be excited, and they are being accepted by the electron acceptors. But here one small thing is taking place, that is the electrons, when they are moving to the cytochrome chain, that time NADP reductase enzymes present there, which is converting the NADP plus H plus to NADPH. So this process is taking place in the case of photosystem 1, that is P700 nanometer, and another process again is taking place, that is in photosystem 2, where we can see the water is being splitted into proton as well as electron plus oxygen. So this electron is being transported to the system back. So this system is a non-cyclic system. It is in the form of Z. It is in the form of Z. Therefore, this system is also known as Z scheme. I think you would have understood about non-cyclic photophosphorylation. We can see it again. Okay. These electrons are picked up by an electron acceptors. Now the electrons are then passed on to an electron transport system consisting cytochrome, which I told you about, cytochrome chain. So electron from the electron acceptor will be accepted by the cytochrome chain. From that chain the electron will be passing and later on it will be passed on to PS1. Since the movement of electrons in this step is downhill, there occurs phosphorylation process, that is ATP is synthesized. So here what is happening when this process is taking place? This is downhill from top to the down area, the electron is moving. So during this process what is happening? ATP is synthesized because lump sum amount of energy is produced and this energy is utilized there for the production of ATP. And next one, simultaneously the electrons in the reaction center of PS1 gets excited on receiving the light of wavelength 700 nanometer. Already I told you when this process is taking place, that time here in PS1 what is happening? The chlorophyll A molecule will be getting excited because of the absorption of light energy, and later on electrons from PS1 are transferred to an electron acceptor and electrons are then transferred to an energy rich molecule, that is NADP plus, to form NADPH2. Okay, the whole scheme of electron transfer from PS2 to PS1 and the formation of NADPH2 is called Z scheme. So that is related to non-cyclic photophosphorylation. My dear children, it is very important. Now number 2 is cyclic photophosphorylation. It occurs in stroma lamina. Stroma laminae lacks PS2 and NADP reductase enzyme. In the case of grana laminae, we have seen NADP reductase enzyme is present there and PS2 system is also present there. That's why there the PS1 as well as PS2 reaction was taking place there. And in the case of cyclic photophosphorylation, it is taking place in stroma laminae and it is lacking PS2 system as well as NADP reductase enzyme. Now what is happening? The excited electrons are cycled back to PS1 through electron transport system. In the case of cyclic photophosphorylation, what is happening? Chlorophyll A molecule will be absorbing the light energy and it is getting excited. So what will happen? The electron will be jumping from the original state, that is the reaction center, and it is being accepted by the electron acceptor, and that electron will be carried by the series of cytochrome chain and again that is being taken back to the reaction center. That is why it is being said the excited electrons are cycled back to PS1 through electron transport system. Phosphorylation occurs due to the cyclic flow of electrons. As the electrons are flowing in a cyclic manner, that time ATP synthesis is taking place and only ATP is synthesized here. But in the case of non-cyclic photophosphorylation, with the ATP, NADPH2 is also synthesized. But in the case of cyclic photophosphorylation, only ATP is synthesized. You can see that is related to the cyclic photophosphorylation process. Okay, my dear children. So I think you would have understood about this much related to photosynthesis. It is very important, but this chapter is very interesting. If you go through the chapter very clearly, you will be able to understand each and every part of that chapter. So just go through the chapter 1 time, 2 times as well as 3 times so that you will be able to understand it. And if you are having any doubt related to this chapter, surely you can give me a call so that I can help you for clarifying your doubts. By saying this, I wind the session here. We will be meeting in the next session. Till then, take care.